to this part B of Tetrad Analysis or gene mapping in fungi. And to understand this part, I recommend you to first see the previous part of Tetrad Analysis, where I have discussed the two different fungal species that are used for most of the genetic analysis, as well as what are tetrads, what are the different types of tetrads that are obtained, and how you can differentiate between whether genes are linked or not linked on the type of tetrads that are there. So in the previous video, we had discussed what are the consequences if the two genes are not linked, and today we'll be talking about what are the consequences if the two genes are linked. So here we are using two nutritional mutants, and these are arginine and uracil. So one strain is a double mutant for arginine as well as uracil. So this strain requires both of these nutrients for growth. On the other hand, this is wild type for arginine and uracil, and as I had told you, that the wild type are written in capital letters and the mutants are written in small letters. So when you make a cross of them, it becomes a diploid cell. Now this diploid cell, before undergoing meiosis, it undergoes replication. So this is a replicated condition. So this is the one homologue and this is the other homologue, both of which have replicated. For simplicity purposes, I have not written the names of the loci because it will make it more complicated. So just remember that this is the loci near the centromere is arginine, and the one that is away from the centromere is uracil. So let us consider first condition in which there is no crossing over. So when there is no crossing over, then we get what the type of tetrad that is obtained is called as parental ditype. So as you can see, all these chromatids they separate, and so you have a parental ditype. Now if there is a single crossing over between these two loci, the type of tetrad that finally we obtain is a tetratype. Tetratype is where you have, this is a double mutant, this is a double wild type, so these are the parental combination, and these are the recombinants. So tetratype, as I had told you, it has 50% recombinant and 50% parentals. Now there's a possibility of double crossover. Now double crossover can happen in many ways. So first we'll consider double crossover between two strands, and these are the two same strands. So there's a double crossover. So when double crossover takes place, ultimately the type of tetrad that is obtained is of parental ditype. So double crossover involving two strands leads to parental ditype type of a tetrad. Now the double crossover between three strands. So these are the three strands which are obtained. So when a double crossover takes place, the tetrads that are obtained are of tetratype, where you have 50% recombinants, 50% non-recombinants. Now on the same thing can happen where you have a double crossover involving the other three strands. Not in the previous one this was not involved, but now this is involved, this is not involved. So again this will lead to a tetratype. Now when a double crossover involving all the four strands takes place, so this is the only way by which we can get a non-parental ditype. So what you see over here is when their genes are linked, so the number of non-parental ditype is very, very less compared to the number of tetratypes or parental ditypes. Now how do you analyze this? So to analyze the distance between the two genes, we use the formula, or we calculate the recombination frequency, which is NPD, that is non-parental ditype, plus half T, which is NPD is recombinant, half T, because tetrads 50% is recombinant, 50% it's not recombinant. So this is half T, and this is divided by total tetrads, okay, and this is into 100. So what we get ultimately is we get the recombination frequency, and as we know, recombination frequency, whatever we get, is equal to the distance in centimorgan. Okay, distance in centimorgan. So this will give you the distance between the two genes. So this is how one can do tetrad analysis. Now, other than finding out the distance between two genes, this kind of analysis also helped to clarify one mistaken model. Initially, when the recombination was identified, so there were questions whether recombination takes place before replication or it takes place after replication. Okay. Now suppose if there's if you have two arbitrary genes A, B, these are the homologous chromosomes, right? Now if the recombination takes place before replication, so what ultimately you will get is you will get the number of non-recombinants will be much more compared to the number of recombinants. Okay, so we will get parental non-parental ditype more than the tetratypes. Okay, so this is a non-parental ditype. So if the recombination takes place before replication, the number of NPD will be much higher compared to other tetrads. But we know that NPD is always much less. So therefore, what it proved was that recombination takes place after replication has taken place. It's not before replication. Okay. Now, as I had told you that recombination or using Neurospora, we can analyze more different types of phenomena. So, this is the chart where I'll be showing you what are the additional information you can obtain by using Neurospora crassa. So, this is Neurospora crassa. Okay. And as you see over here, this is after meiosis 2, there are 4 spores that are present, and these 4 spores, however, it undergoes mitosis to give rise to an octad. So Neurospora crassa ultimately gives rise to 4 haploid spores. Also you can see over here is that the ascus is very elongated structure, and the diameter of the ascus is such that spores can only move in one direction, they cannot cross each other. So as a result, if this is a diploid cell, so this is the spindle fiber, how it is formed, it gives rise to 2 cells. Now the spindle fiber forms in this manner, it gives rise to 4 cells, and so these 4 cells, they undergo mitosis to give rise to 8 cells. So these two cells over here, they are actually derived from this haplospore. So therefore they are identical, right? Now, if what one can understand, additional information that can be derived out of this is, one can find out using these octads the distance between the centromere and the gene, right? Now I have taken an arbitrary example in which one of the loci is B, which will give rise to blue spores, and the other loci is red, which will give rise to the red spores. Now if there is no crossing over here, then what we get is, we get all the blue spores either on this side or on this side. So this is the pattern which we get, and this is called as the separation of alleles takes place in first meiotic division. Okay? Or the segregation of alleles if they take place in the first meiotic division. So this is the pattern which we get, and if you just draw an imaginary line in the center, you will see all the 4 spores of same kind on one side and all 4 spores of same type on the other side. If the orientation of these chromosomes are reversed, you will get this feature. So if you have octads which have this kind of arrangement of spores, you know that the 2 alleles, they have separated in the first meiotic division, right? Now suppose if there is a recombination which has taken place between over here AN, between the centromere and this G. So a recombination, a crossing over has taken place. So what you get over here is, you get these resultant tetrads, and then when they undergo mitosis, you see these are the 4 ways by which they can be arranged. So this pattern tells you that the separation of alleles or the segregation of alleles has taken place in the second meiotic division. Okay. Now, the second meiotic division, if you get this kind of a pattern, again if you draw an imaginary line from the center like this, so you will notice that this is different from the first division segregation pattern. Okay. Now, by analyzing the number of second division segregants or the tetrads which show second division segregation pattern, one can identify or one can find out the distance between the gene and the centromere. So the distance between the gene and the centromere can be analyzed by looking at the second division segregant pattern, and the formula is you have half of all the second division tetrads divided by total number of tetrads into 100. So this will give you the recombination frequency, and this can tell you the distance between the two genes. So this is how one can identify whether the two genes are linked or not by looking at the tetrads and the distance between the gene and the centromere. Okay. Now, if you have a numerical problem, and this problem where you have been given a cross between, let's say, A, B, okay, and you are given a group of all the tetrads, what are different combinations that are obtained. So now the questions, two questions you are asked. The first question is, are these two genes linked? That you can find out by looking at the NPDs and PDs. Second is, if these two genes are linked, what is the distance of these two genes from the centromere? So there's possibility that the centromere is on one side and all the genes on one side of the centromere. The second possibility is the centromere is in the center and you have this arrangement. To solve these kinds of questions, what you have to do is, first you have to find out the distance of one loci from the centromere. So what is the distance of A from the centromere? You can calculate, you can find out the distance. Let's say this is X centimorgan. Then you have to find out what is the distance of B from the centromere. So this is, let's say, Y. Distance of B from the centromere is Y centimorgan. Now, if you find out the distance between A and B by doing this kind of using this formula, you can find out the distance between A and B. So if the distance between A and B exceeds the distance of A, this as well as this, so you know that A and B are on two sides of the centromere. So what I am saying is, while doing this analysis, if you find that the distance between A and B is much more compared to X independently or Y independently, or in other words, if the distance is a summation of X and Y, you know that the two genes are on the two sides of the centromere. If the distance between these two is less compared to any of this, then you know that it is the arrangement is like this. So whenever you have a question, what you have to do is, you have to first find out for each loci the distance between that loci and the centromere. So for that you have to analyze the tetrads. So first you just look at one loci. Whether if, okay, just an example, I'll tell you. If you have, let's say, the arrangement is like this, A, right? Now, mostly the octad, the whole entire octad will not be given, only the tetrad will be given to you. So if the arrangement is like this, so you know that this is the first division segregants. If the arrangement is like this, you know that these are second division segregants. So by calculating what are the number of second division segregants, you can find out the distance between centromere and this A loci. Similarly for B, you will have in this manner. Let's say if this is this, it's like this. So this is first division segregants. Second division will be This will be second division. So by analyzing the first division and second division segregates, one can find out the distance between the two genes, and then one can solve the numericals in this manner. So these are the things that you have to just remember. Just go through this chart and see how you're getting the different tetrads, the formula, what is the importance of Neurospora in identifying the distance between gene and centromere between the two genes, and so this is done. So this is about tetrad analysis, and if you understand this, you'll be able to solve the numericals that are given to you. Thank you.